Hello students, today we are going to discuss about bacterial genetics and modes of gene transfer in bacterial cells. So bacteria can transfer genes among themselves in three possible ways. Number one is transformation that involves the uptake of free DNA molecules released from one bacterium, that would be a donor cell, by another bacterium cell, that is a recipient. No cell-to-cell contact is needed when bacteria exchange genes through transformation. We would look into the details of transformation a bit later. Conjugation involves direct transfer of DNA from a donor cell to a recipient. This is the type of gene transfer where cell-to-cell contact is mandatory, and keep in mind, conjugation is always a one-way exchange. Only one cell at a time acts as a donor. In transduction, bacterial genes are carried from one cell to another by bacteriophages. Here in this diagram you can see all these three possible modes of gene transfer: bacteria absorbing free DNA molecule from the environment, a cell-to-cell contact called bacterial between bacterial cells called conjugation tube is present, one cell is acting as a donor, the other cell is a recipient cell, and a bacteriophage is carrying a bacterial gene from one bacterial cell to the next. Other important thing is that not all these processes are present in all species of bacteria. For example, Escherichia coli does not contain genes that encode for the proteins required to take up free DNA. So in case of Escherichia coli, genes can only be transferred through conjugation or transduction, but not through transformation. Why? We'll look into that. So number one, transformation. The process of transformation was first discovered by Frederick Griffith in 1928 when he was actually working on a bacterium, Streptococcus pneumonia, and was trying to make a vaccine for disease, respiratory tract infection, pneumonia. Streptococcus pneumonia has two strains, type S and type R. Type S is a virulent strain, a disease-causing strain. Type R is non-pathogenic, non-virulent strain. Difference between the two strains is ability to form a polysaccharide capsule, which is present in case of type S but absent in type R. Due to this coat, the type S colonies appear smooth under microscope, so the name S comes from smooth and R comes from rough colonies. A capsule may be of several different antigenic types like type 1, 2, 3 and so forth. This classification is based on specific molecular composition of polysaccharide coat that definitely depends upon the genes, depends upon the genotype of the cell. If type 2 cells are injected into bloodstream of rabbits, the immune system of rabbits will produce antibodies that react specifically to type 2 cells. They will not react with type 1 or type 3 cells. It indicates that all these polysaccharide coat types are immunogenically different from each other. They have different chemistry, you can say. What was the Griffith's experiment and its findings? Let's first look at this diagram over here. First of all, Griffith inoculated live type 3S bacteria into mouse. The mouse fall ill and ultimately died, and type 3S bacteria were discovered from his body. Next, he inoculated heat-killed type 3S bacteria into the mouse. Mouse remained healthy. It means type 3S bacteria, when they were heat-killed, they lost their ability to cause disease. Similarly, type 2R bacteria, when inoculated live, you can see over here, they lack polysaccharide coat, which was shown red in previous steps. Here you can see the polysaccharide coat shown red here. Live type 2R bacteria, when inoculated, fail to cause disease and mouse remain healthy. Last step, you can see when heat killed type 3S bacteria and living type 2R bacteria were inoculated in the form of a mixture, not only the mouse died of the disease but also type 3 live bacteria were discovered from the dead body. We have inoculated heat killed type 3S, but he recovered type 3S live bacteria from the mouse. What can you suggest has happened during this experiment? The type R cells that contain a mutation in their gene for polysaccharide coat can mutate back to type S cells, but if type R cells have encountered a backward mutation, we should have recovered type 2S bacteria, not type 3S bacteria. Yes, but in actual experiment, the Griffith recovered type 3S bacteria, not type 2S bacteria. It means no backward mutation has occurred, but a component of dead type 3S cells has somehow converted type 2R cells into type 3S cells. This component, which was later confirmed that it was a DNA fragment, was named as transformation principle. So here from this experiment you can conclude that transformation occurred when live type 2R cells were grown in the presence of heat killed type 3S cells. Griffith's experiment demonstrated that type 3S phenotype of the transformed cells was not only created but also passed on to the progeny. That was due to permanent inherited change in the genotype of the cells. So when these cells replicated, their transformed genome was inherited to the next progeny. Live type 3S bacteria for this particular reason were recovered from the dead body of ill mouse. Let's now discuss mechanism of transformation. Keep one thing in mind, not all bacterial cells can absorb free DNA from the environment. Only the cells we call competent cells can absorb free DNA from the environment, and these competent cells have certain genes that are encoding proteins for this process. These genes are called competence genes and their products are called competence proteins that make these bacterial cells capable of taking up free DNA. The process by which cells become competent is understood with the example of Bacillus subtilis, where small peptides called competence pheromones are secreted by cells and accumulate at high cell density at late stages of their growth. High concentration of these proteins induce expression of genes encoding competence proteins. Here in this diagram, you can understand the process of transformation and role of these competence genes and their products competence proteins in the absorption of free DNA from the environment. Here at the surface of bacterial cell, there is a cluster of proteins present. You can zoom into this cluster here in this diagram. First of all, cell wall of the bacterium contains competence G protein, another one competence EA protein. What do these proteins do? These proteins actually bind free double-stranded DNA from the environment and convert into single-stranded DNA. Another protein, COM-EC protein, competence EC protein, acts as a channel protein through which this DNA will pass the cell membrane and enter into the cytoplasm of bacteria. But along with competence EC protein, there is another protein called deoxyribonuclease. Deoxyribonuclease, while this double-stranded DNA molecule is crossing the plasma membrane, this enzyme will degrade one of the two DNA strands. You can see over here is being destroyed, is being degraded into individual nucleotides, and the other strand passes through this channel protein competence EC into the cytoplasm of bacteria. What makes the other strand immune to this deoxyribonuclease enzyme? Actually, these are single strand binding proteins. These proteins protect the other strand of the DNA from deoxyribonuclease enzyme. The next member of this competence proteins is recombination A protein. Recombination A protein binds to the remaining DNA strand of received molecule. The recombination A protein incorporates this received DNA strand into the host genome at complementary site. And during this incorporation, the equal piece of DNA strand is removed. So you can say this is a recombination process. One strand of host genome is removed and the received strand here shown in yellow is incorporated into its place, and a heteroduplex is formed, a double-stranded DNA carrying one strand of one original strand from the recipient cell and one strand of the donor cell, which has been incorporated in the main genome. When this bacterium will divide and copy its genome, the one cell will receive DNA molecule containing, you can say here, A minus gene. The other cell will receive this outer strand in duplicated form containing A positive gene. So here, through transformation, A minus bacteria has been transformed into A plus bacterium through transformation, and this characteristic is passed from one generation to next generation. So this is the process of transformation. We will look at the other two processes, conjugation and transduction, in our next video. Thank you.